Yo, and welcome to Shooting the Soil. Today, we're looking at my sorghum, or some soil, I should say, under my sorghum, that has developed a fungal pathogen. Anthracnose is, I believe, what it's called, unfortunately. So, we are going to look at the soil and see what may have caused this, or how we can improve it and essentially prevent it from happening in the future. So, one of the best ways to compete against fungi, or fungal pathogens, I should say, is actual beneficial fungi, because they, in the microbiome, compete against each other. Which is kind of ironic, because the first thing we go to when we see a fungal pathogen is a fungicide, which is indiscriminate and kills all fungi, preventing what little you may have had in your soil to prevent it in the first place. So yeah, to reiterate, what we want to prevent fungal pathogens, like this anthracnose and other things like powdery mildew, is actual good beneficial fungi. So we took a sample right under the infected sorghum, and we're seeing some fungi strands like this, but not exactly what we would like to see. I mean, it's a decent amount. We're seeing another little piece up here. We are at 100x magnification. So basically, we are looking for signs and what we could possibly improve on this soil to prevent fungal pathogens. Seeing some good aggregation over here. That's a nice sign. No cause for concern, but we're probably going to have to zoom in. Oh, there's a bubble. We got anything else up here? That's an interesting. I almost thought that was microplastic here. This is inside of a bubble on the slide. The water's over here. That's just a piece of mineral floating around. But we could see hyphae here and here. So that's an odd looking fungal strand, just how it's blue like that. Huh. That's interesting and something that you don't normally see in the soil, and it's very large. Like your normal fungal strands are this size, the dark brown ones, the good beneficial ones. And the endophytic mycorrhizal can be even smaller than that. Like your arbuscarizal mycorrhizal can be really tiny, because it forms those symbiotic relationships with the plant and goes right into the roots. Oh, actually, we got some of the more of that blue stuff actually in water over here. Huh. I have not seen a fungal strand that color. That light blue. And it's the first time I've seen this fungal pathogen. May just be a coincidence. Alright. Man, probably should go into 400x to inspect what's going on here a little bit closer. But we're going to continue zooming around. So, this is looking a lot like clay. When I did the sample, we had a real good amount of nice looking soil on top with organic matter. Hence, I've remediated this bed. It's a fairly new bed that I made. It's not very raised, but raised just a little. And I put down a whole bunch of leaf mold and compost on top, and kind of let it went wild to, let the plants feed the soil and the microbiology to build soil. But other than that, I haven't really done that much else. Alright, so we see a good, nice, large piece of organic matter here. We can see that this was once plant matter. That's a good sign. But anyway, when I'm taking the sample and I get down below the compost and everything that I got, and I tried to get a good happy medium of both the nice fluffy looking stuff, mostly my compost on the top, and what was down lower, which was really kind of like grayish clay, I would have to say. Which usually my clay is very orange around here. And this clay was gray. So that might be a sign of it just being wet and anaerobic, I'm thinking. Because, yeah, we're not too far above sea level. Actually, where this bed is, it's probably about 7 feet above sea level. This big pink thing, I would think this is plastic. That doesn't look like any kind of fungal, bacterial, any kind of strand. I'm betting that that's plastic. But as you notice as we zoom around, like we see one little piece of fungi there. This is organic matter. This is probably a root hair. But there's one little piece here, but very little fungi. Yep, some more organic matter here. But if we want to prevent things like powdery mildew and other fungal pathogens, like that I have here on my sorghum, we need closer to a 1-to-1 fungal to bacterial ratio. And on that note, let us zoom in to 400x so we can actually see the bacteria. So we can actually get an idea of the ratios that we are talking about here. Alright, let's get focused. Oh, where we go? Don't bump into the side, please. Come on. Did I go past it? Oh, there it is. Whoa. Alright, let's put a little shading on this so things can get a little bit more in focus. Yeah, maybe right about there. Might be a little bit too much. We can increase our lighting so you guys can see more. Alright, so we see a whole lot of bacteria down here. Definitely no shortage of bacteria, which isn't very uncommon. Like, actually, some of the, what I would consider, more basic soils, lesser succession soils, actually seem to have a whole lot of bacteria, surprisingly enough. Sandy soils, what have you, bacteria really multiplies first. But what we're looking for is the diversity. We need the whole ecosystem to benefit and feed our plants. And give them what they need to have their ultimate health so they don't get fungal pathogens. And it also helps that the good fungi will compete against the bad fungi and prevent them from happening in the first place. Alright, so zooming around at 400x now. Yep, a lot of little pieces in there. Yep, you can see in that clay that I moved in there, that gray clay that was about 4 inches down in this soil. All those little clear pieces. Just small clear minerals that don't do much for the soil. Eventually they'll be broken down and aggregated and turn into these ones that have glomulin on them for the bacteria that are slowly reducing, and they'll eventually form aggregates. But yeah, when was the last time you saw a fungal hyphae? Oh, there's one. Little piece. nice brown looking one, but far from a, I would say, I don't know, what would we give this to a fungal bacterial ratio? Whole lot of bacteria. And a fungal, oh, there's one. Every 1, 2, 3, 4. Yeah, 1 to 5. 1 to 6. Oh, did we have one here? All right, yep. 1 to 6 it is. That's our rough estimate. Because essentially what you want to see at 400x is you want a good fungal, or at least a piece like this, if not a big fungal strand, in every single frame at 400x. That's when you know you're at a 1-to-1 fungal bacteria ratio. With having as much bacteria as you see down here right now, that would be. Which is, I mean, this is high average. Yeah, actually, where was? We had a couple of good, nice fungal. I just want to take a short little video here. Excuse me while I just do this. Alright, that ought to do. Need to do that. Doing a collab with Guardian Like a Viking. If you guys haven't checked out him on YouTube, I would highly suggest it. Because, yeah, he's one of the most experienced gardeners I have seen on YouTube. I really like what he has to say, and he's taught me a lot and helped me to have a little bit more of a green thumb. Never really had one in the first place, but we do our best. And the microscope has probably been the best thing that's helped me out, to be honest. And, oh yeah, I wanted to mention the fungal spore that we see right here. Another piece of little fungal hyphae. But yeah, a whole lot of bacteria. I'm surprised we haven't seen any funky strains yet. Strands, I mean. I was suspecting to see like actinomycetes, omycetes, actinobacteria, because those are the things that really lead to all sorts of fungal pathogens. Well, mainly in my experience, powdery mildew. What's this strand that we have here? Oh, there we go. See, yeah, this is what I'm talking about. So you see how it's just not uniform. It gets bigger here. We don't see any fungal hyphae. So this is what I'm talking about as a funky strand. It's hard to tell if it's a bacterial strain or possibly an omycete, or it could just be a saprophyte that got all wrapped up while I was taking the sample and shaking it about. But since we don't see any separations in the cell walls, like that's, can I get a little bit? Nope, that's just the shadow from this piece on top of it. So it's really helpful to go in and out of focus to tell that sort of thing. And yeah, no separations in the cell wall, not very uniform, funky strand. Not quite sure if it's an omycete or actinobacteria, but it's not a good one. And that's just one of the signs that could lead to my fungal pathogen that I have on my sorghum. Plus, when I was doing my research on it, I did find unique and rare types of sorghum are, the hybrids are very acceptable to this type of fungi, especially when they're not grown in their native habitat. So that makes a lot of sense, yeah. That they were brought quite a ways away from where I bought the seeds. We got a fungal spore right there. I mean, there's fungi in there, but yeah, it's not where we want it to be, or evidently it's not where it needs to be to outcompete. Oh yeah, I would say Hank Derbs, I would say 1-to-1 is definitely our goal for that plant. That's it's more of an advanced plant, and yeah, 1-to-1 is pretty much always your goal unless you're growing something like a brassica or a leafy green. Then you might be want to be more on the bacterial side. But other than that, pretty much you always want a 1-to-1 fungal to bacterial ratio. Alright, where are we on this slide? I don't think we've gone down to the bottom yet. We keep on coming back up and circling around. Let's just kind of, yeah, we're just going to go all the way down here. Skip around, because we haven't looked down here at all yet. Oh, yep. Another funky-ish looking strand. Well, ultimately super ununiform, but clear. And no septums. No septi. There's a good one. Scroll along. A lot of clear bits in here. We need more aggregation. Aggregation is one of the best signs of biological activity. We're not seeing many clumps. So let's find a clump. So here's a clump. Yeah. That's thus all that gold brown, those humic acids, but it's not super clumped, you know? We want to see all clumps and not all this clear stuff. But we'll get there. We will get there eventually. That looks like that kind of looks like a diatom. I am right by the ocean, and I do add a lot of kelp, so it would explain why we would see diatoms in the soil, which generally aren't seen in there. They can be seen, but definitely more likely if you add, if you're away from the ocean, you probably add a kelp product of some sort to get them in there, or fish hydrolysate. So yep, seeing more weird strands. Let's see if I can get a better focus on that. So that's almost looking like a bacterial strand. Just possibly not. I'll try a little bit more shading. What if we go a little bit darker here? Maybe we can get a little bit more definition. Bump up my ISO a little bit. And a little bit better. But yep. Need to outcompete those with more good fungi. Here's another very ununiform, not so beneficial, but then we got a decent looking one there. There's hope. Oh, and here's more hope. Big fungal spore. Those big football shapes are some of my favorites for the fungal spores. Here's another one. And a whole lot of bacteria. I think this all bunch of organic matter over here. Yep. Little piece of a leaf there. breaking down, providing food for the microbiology. Yeah. And a good fungal hyphae there. But they're few and far between. Here's the edge of the slide as drying and moving in. Alright, moving across the middle here. Probably take one more scroll across. And no amoeba. I have not seen one amoeba yet. That's interesting to me. Oh. Another omicete kind of looking strand. Oh, and as I say that, I just spot one. Pretty sure that's an amoeba. Yeah. So we do have a amoeba in there. That's the first one I've noticed. Usually I see a whole lot more. But that's because we probably only have about 3 inches of good soil and then it hits clay in this bed. So we gotta build it up. Here's a cool looking fungal spore. I always like to see those ones. Yeah, we do have some good fungal hyphae. I'm surprised I saw this much fungal hyphae in there. But we have done a bunch of fungal inoculants. I just don't think it has time to take hold, or it's just in the upper layers and we're getting infected from all the down below where the good fungal hyphae hasn't taken over yet. Because we've been using some Rootwise and some mycogenesis from Advancing Eco Agriculture. So that's probably the only reason why we're seeing, some of it's from nature, but probably most of it is because I've done these inoculants. Because if I just go out and sample the, just regular clay or, topsoil onto the grass, we actually don't see as many fungal hyphae as this. Only if I go into the forest next to my house where there's mushrooms popping up all over the ground and it hasn't been touched in probably about 100 years. But before that, you can definitely tell that it was a farm field, my property. So it's not a very advanced forest. It's a young forest, we should say, but we got a couple, 80 to, or I would say 60 to 80-year-old oaks. Nice, big round ones growing on the property. But anyway, alright, we're kind of just circling around, hoping to see some more interesting stuff. And, so here's an example of a saprophyte. So you can see how I can definitely make out a septi there, and I can make a septi there. It's very clear. Could actually be some other type of fungi, but just it's very young and hasn't filled up with nice color like this one over here because it just hasn't eaten much. It's just started growing. But most likely, it's a saprophytic fungi, most commonly found in, breaking down organic matter in your compost. It's that white fuzzy stuff that grows in your compost bin if you don't take it out too often. But, yeah, so that's actually not all that bad of one. And you can see it got kind of roughed up over here and sort of looks ununiform. But it's pretty much the same width all the way through. And yeah, let me point out the very hard to spot septi, but we can see one right here, and I noticed another one, I think, right up over here. Yeah. So yeah, it's a couple of separations, so we know it's fungal. We know it's a fungi strand. Yep. Another one right over there, so it's either a very young or it's a saprophyte that usually don't get this dark brown color like this, like these ones over here. Juglone. I've never heard of juglone. I haven't heard of black walnut trees. Um, Definitely being a problem for growing other species around them is yeah. Can you, what exactly is a juglone though? A bacteria that eats juglone. Bacteria does eat a lot of stuff, and there is so many different strains. We can only see under the microscope three different, basically, shapes, metamorphic shapes, is the only identifying properties we have with the microscope. And there is, I think, I want to say millions of different types and genomes of bacteria. It's pretty wild. And yeah, I know some can even eat plastic, so I wouldn't doubt it. But as far as identifying them under the microscope, that would be, yep, you would need DNA sequencing or something like that to be able to identify them. That was another question from Hank Derbs asking if bacteria eats juglone, the toxic stuff from black walnut trees. Yeah, I didn't know that. I knew black walnut trees definitely compete and make the soil very acidic. So, is the juglone what one of the things that causes the acidity? That's what I'm guessing anyway. But well, this slide is drying up here, so we may call it for the night, unless anybody has any more questions. But yeah, definitely need more aggregation. Well, as we come into a big aggregated area, but that's kind of because of it. What was that? Was that a diatom? No. Slides drying up and pushing things together here. Wonder if that's plastic or some kind of weird spore. It's very glowy. But anyway. Yeah, it's very acidic, says Hank Derbs. The juglone. Yep, definitely makes black walnut. Like, my dad gave me a couple black walnuts, and I wanted to find a spot to plant it on the property, and I'm just like, I just got no spot. I might want to grow other plants here. I can't do it. Or the walnuts dropping on cars too. You don't want to plant it over a driveway. That might be another amoeba there. Yeah, I think I see a cell wall. So yeah, that's another thing. There's only two amoebas. I think we only saw two amoebas on this whole slide. That's a rarity. I really think I'm starting to think amoebas are one of the best things you can see. It's just a sign of good soil. It's just a good sign. In my opinion, in my experience so far. Alright. So yeah, definitely need more aggregation. Willing to bet the fungi came from Yeah, just because I only got 3 inches of good soil in this bed, and after that it's clay. It's really low. It's staying wet. And I'm like, oh, it's sorghum. It's grass. Like, let's water this more than the rest of my garden and make the grass go really quick. That might have been a mistake. We might have made that lower clay stuff a little bit anaerobic and made a perfect breeding ground for whatever the fungal pathogen that or the yeah, let me make sure I copied and pasted it. Let me make sure I'm saying this. Yeah, Anthracnose. Yeah, Anthracnose is what it looks like that's growing on my sorghum. Well, I think we learned a little bit. Definitely gonna. I've already put down a little bit more mycorrhizal inoculants to hopefully outcompete it. I also sprayed some LAB on there because I feel like that. Well, I know that does really good against powdery mildew, so I figured might help against this. But I will let you know, and I'm also going to do a little bit more research. So hit that subscribe and bell notification because I'm going to. There is one sorghum in the whole bed, way in the corner, that is actually not affected, and it's twice as tall as all the rest. So I'm going to do some in-depth sampling of the two and see if I can put together a compare and contrast video for you guys and learn a little bit more about why just one of the sorghums doesn't seem to be infected by the anthracnose. So anyway, stay tuned for that. And yeah. We will catch you on the next one, and happy growing until then. And yeah, all right, have a good night, guys, and we will catch you on the flip side.